Ever wondered how our genes express themselves? Well, it's through a fascinating biological process called gene expression. This process is like an intricate dance of molecular interactions where the genetic information within our genes is translated into functional outcomes. In simpler terms, it's how the instructions contained in our DNA are used to create the proteins that make us who we are. Gene expression involves two main stages: transcription and translation. Picture them like this. Transcription is akin to a scribe copying a manuscript where the original text is DNA and the copy is a type of RNA. This copy then serves as a guide in the next stage, translation, which is like a craftsman using a blueprint to build a structure, with the blueprint being the RNA and the structure being a protein. Now that we have an understanding of what gene expression is, let's delve into how it happens, shall we? The first stage of gene expression is called transcription. It's a fascinating process where the genetic information stored in a segment of DNA is copied into a molecule called messenger RNA, or mRNA for short. You see, our DNA is like an immense library of blueprints, and these blueprints need to be copied onto a portable format so that they can be read and used elsewhere. That's where the enzyme RNA polymerase comes into play. Acting as a diligent librarian, RNA polymerase unzips the DNA double helix, revealing the genetic code inscribed within. RNA polymerase then meticulously reads and copies one strand of the DNA, synthesizing a complementary RNA strand. It's a bit like copying a recipe from a cookbook, but instead of ingredients and cooking instructions, we have nucleotides and genetic codes. This new mRNA strand is a mirror image of its DNA template, a perfect copy of the genetic information. Once the transcription is complete, the mRNA strand detaches from the DNA. It's now ready to embark on its journey, carrying the precious genetic information out of the nucleus and into the cytoplasm of the cell. And with that, we have completed the first stage of gene expression. This process, as intricate as it is just the beginning. But where does this mRNA go and what does it do? That's where the second stage comes in. Stay tuned to find out more. The second stage of gene expression is known as translation. This is where the magic happens, where the genetic information encoded in mRNA is put to work to create something truly tangible, a protein. Now let's dive into the process. Our protagonist, mRNA, carrying the genetic code, binds to the ribosome, a molecular machine that will facilitate the protein synthesis. The ribosome recognizes the start codon, AUG, on the mRNA, marking the initiation point for translation. Enter our supporting cast, the transfer RNA molecules, or tRNAs. Each tRNA carries a specific amino acid and has an anticodon sequence that matches with the corresponding codon on the mRNA. The ribosome moves along the mRNA and each codon is matched with the appropriate tRNA anticodon. The amino acids carried by the tRNAs are then joined together in a specific sequence, forming a growing polypeptide chain. This chain is the primary structure of the protein. The translation process continues like a well-orchestrated dance until a stop codon, either UAA, UAG, or UGA, appears on the mRNA. These stop codons don't code for any amino acids. Instead, they're like the director yelling, "Cut!" They signal the end of the translation process. And that's how a protein is synthesized. But the story doesn't end here. Like any good story, there are twists and turns even after the main plot has unfolded. But we'll delve into those in the next scene. Stay tuned. After translation, proteins undergo various post-translational modifications. You see, proteins are not just a simple chain of amino acids. They're intricate, complex structures, and they need a little extra work before they're ready for action. This extra work involves folding the protein into a specific three-dimensional shape. Think of it like origami, but on a molecular level. This shape determines the protein's function, whether it's to fight off invaders, carry messages, or build structures within the cell. But that's not all. Some proteins also need additional functional groups. These can be as simple as a single atom of phosphorus or as complex as a carbohydrate chain. These additions can help the protein interact with other molecules or change its behavior in response to the cell's needs. Lastly, some proteins need to be trimmed down to size. Just like a sculptor chiseling away excess marble, enzymes in the cell can cleave specific segments of the protein, activating it for its specific role. With these final modifications, our protein is now ready to perform its function in the cell. So now we have a good understanding of how our genes express themselves. We've delved into the two key stages of gene expression: transcription and translation. Transcription, where DNA is converted into mRNA, and translation, where mRNA is used as a blueprint to create proteins. We also touched on post-translational modifications, which fine-tune and activate these proteins. Remember, every protein in your body went through this process, a testament to the sophistication and precision of life at the molecular level.